For the investigation part, we will carefully perform the practical to observe the structure of a seed. Before starting, it is important to follow some safety precautions. Handle the scalpel or blade with care to avoid cuts, always cutting away from your fingers and body. Use forceps rather than your fingernails to remove the seed coat, or tester, to prevent injury. Make sure to work on a stable surface, such as a dissecting tray or a white tile, to avoid slipping. Do not run or distract others while using sharp instruments. After completing the practical, wash your hands thoroughly to remove any residue from the seeds or equipment. Following these precautions ensures safety while carrying out the investigation. For investigating the structure of a seed, the objective is to carefully examine and describe its parts, with particular focus on the seed coat, or tester, and the embryo, including the radicle, plumule, and cotyledons. During the investigation, observe the position of the radicle, the micropyle, the hilum, and the tester. Start by looking at the external appearance of the seed to identify the radicle, plumule, and cotyledons. Then, carefully remove the tester to study the internal structures more clearly. Finally, remove one cotyledon to get a detailed view of the embryo inside. For this investigation, the materials and apparatus required include a soaked bean seed, forceps, a scalpel or blade, a dissecting tray or white tile, and a hand lens. The soaked seed is used because it is soft and easier to dissect, allowing clear observation of both the external and internal structures. The method begins by placing the soaked seed on a white tile or dissecting tray. Start by carefully observing the external structure, noting features like the tester, hilum, and micropyle. Using forceps, gently remove the outer covering, the tester, instead of fingernails, to avoid damaging the delicate embryo inside. After the tester is removed, split the seed into two halves along the plane between the cotyledons. This exposes the internal structures, including the radicle, plumule, and the cotyledons. Finally, use a hand lens to examine these parts in detail, making sure to identify the radicle, plumule, and cotyledons clearly. This careful approach ensures that both the external and internal features of the seed are observed accurately, providing a clear understanding of the seed structure and the role of each part during germination. From the observations, we can analyze the structure and function of the different parts of the seed. Externally, the seed is covered by the tester, which protects it from damage and prevents it from drying out. We also notice the micropyle, a tiny opening that allows water to enter the seed to start germination, and the hilum, the mark where the seed was attached to the fruit. When the tester was carefully removed, the internal structures of the seed became visible. The embryo consists of the radicle, which is the first part to grow and develops into the root, and the plumule, which develops into the shoot. The cotyledons act as storage organs, providing the necessary nutrients and energy for the growing embryo during germination. There are two types of cotyledons. Monocot seeds have one cotyledon, as observed in grains like maize, while dicot seeds have two cotyledons, as seen in beans and peas. In monocot seeds, the single cotyledon stores food and helps in transferring it to the embryo, whereas in dicot seeds, the two cotyledons often surround the embryo and directly store the food needed for germination. By removing one cotyledon, we could observe the detailed arrangement of the embryo parts more clearly. From these observations, we can conclude that the tester, cotyledons, radicle, and plumule are all specifically adapted to protect, nourish, and support the growth of the new plant. So, from this investigation, we can conclude that a seed is made up of an outer protective layer called the tester and an internal embryo consisting of the radicle, plumule, and cotyledons. The tester safeguards the seed from damage and drying out, the cotyledons store food for the developing embryo, the radicle grows into the root, and the plumule develops into the shoot. These structures are perfectly adapted to ensure the embryo has protection, nutrition, and the ability to grow into a healthy seedling under favorable conditions. This practical investigation helps us understand the structure of a seed and the role of each part in plant development. It provides hands-on experience in observing and identifying the protective and nutritive adaptations of seeds, which is essential for learning about germination, seedling growth, and overall plant reproduction. By comparing monocot and dicot seeds, we also learn how seed structure varies between different types of plants and how these adaptations support successful germination.